Okay then. So as we know, today's our lecture number 3. In today's topic, we will start with the significance of mitosis and then cytokinesis. Okay?
Yes,
Yes,
ma'am.
ma'am.
Okay. So in the time, significance of mitosis, and then we will start with meiosis and its phases. So firstly, we can have a quick recap of the last class, what we have done in the previous class. Can you just tell me the answer of this question in the chat section? Try to solve this question. This question is from mitosis, which we have done yesterday. Try to solve. Namrata is saying A and Anushka is saying D, but with some little confusion. Harshida is D, okay. Why D? We are talking about mitosis, not for meiosis. Keep it in mind. We are talking about mitosis, not meiosis. In mitosis, there is no crossing over. So why are you saying answer is D? Right. Just remember, the first phase was prophase, in which condensation of DNA occurs. Then second one is metaphase, in which all chromosomes get itself aligned at equator. Then centromere split and move to the opposite pole. Keep it in mind. This is a very simple question, bachcho. Should I answer the question?
Yes, ma'am.
If I say the correct answer is one, exactly. The correct answer is one. Why? In the first phase of mitosis, condensation occurs and nuclear membranes start disappearing. In metaphase, all chromosomes get at equator at metaphase plate, and then centromere split, chromosomes separate and move to the opposite pole, and the last phase is telophase. Right or not?
Yes, ma'am.
Yes. Agree? Why the correct answer should be A?
Yes, ma'am.
Yes. Very good. Next question. A stage of mitosis is given. You need to find out the correct stage and their characteristics. Students who are late, please try to join at exact timing. Your class just start at 5:45. So you need to join at 5:45 only. You're 10 minutes late. Tell me the correct answer. Very good, Harshida. Hurry up, bachcho. Stage of mitosis is given. You need to find out the correct stage with its correct characteristic. We all know all chromosomes are aligned at the equator. That's why this phase should be metaphase. In this point, we can ignore option C and option D. Right? In the option A, they are saying spindle fiber attached to the kinetochore. Centromere split. Centromere split is the characteristics of which phase?
Anaphase.
Anaphase. So this should not be the correct answer. The correct answer should be
B.
very good. Next question. During the metaphase stage of mitosis, spindle fiber attached to chromosome at very good. The correct answer is A. They get attached with kinetochore, not both. Next and the last question. After that, we'll start with a new topic. Same question. Kinetochore. There should be no confusion. Okay then. So we can start with our next topic. In the last class, we have done with the telophase stage. What in the last class, we are done with this phase in which a cell has two nucleus. Now we need to divide its cytoplasm, prophase, metaphase, anaphase, and telophase. All these four phases were of karyokinesis. What do you understand by karyokinesis?
Division of nucleus.
Karyokinesis, division of nucleus. So in the last class, we have finished with the division of nucleus. So after dividing the nucleus, we need to divide its cytoplasm also.
Yes, ma'am.
By dividing the cytoplasm, the cell will get divided into two parts. So we need to divide its cytoplasm. And then we can have two different cells. So cytokinesis is basically the process of division of.
Cytoplasm.
Very good. So in cytokinesis, it can be different for different cells. What is the difference between animal cell and plant cell? Tell me a basic difference. Basic difference between animal and plant cell.
In animals, there is no cell wall. In plants, there is a cell wall.
Exactly. There is a difference of cell wall. In animal cell, we do not have any cell wall. But in plant cell, we have a cell wall, which is made up of cellulose. And that cell wall is inextensible. We cannot break the cell wall. It's not easy for us to break the cell wall. So how we will do cytokinesis in plant cell? In animal cell, it is quite easy to divide the cell into two parts by the process cell furrow method. But in plant cell, we will divide the cytoplasm with the help of cell plate method. You need to write down the method's name. Question can be asked in animal cell how cytoplasm divide itself, or we can say by which method cytoplasm get divided to two parts, with the help of cell furrow method, and in plant cell plate method. Done?
Yes, ma'am.
Everyone, Mishpa, Anushka, Anusha, Rupali. I need everyone response.
Yes, ma'am.
ठीक है. So we can start with next topic. The next one we have significance of mitosis. So Rupali, can you tell me significance का मतलब क्या होता है? Rupali, what do you understand by significance? Anusha, Namrita,
Main characters.
yeah, Kalpana,
Yes, ma'am.
what do you understand by significance term?
Mm,
Yes, Vimesh, आप बताओ.
Importance.
Yeah, we can say importance. Anything else? Ma'am, the process can also known as centripetal. Exactly, Anushka. We can also call it centripetal growth. Cell cleavage method भी हम उसको बोल सकते हैं. It can be also called as cell cleavage method. तो इन द प्रोसेस ऑफ सिग्निफिकेंस, सिग्निफिकेंस का मतलब होता है कि हम उसमें हमें हमारे लिए जो माइटोसिस प्रोसेस है, माइटोसिस प्रोसेस का इंपॉर्टेंस कहाँ कहाँ पे हमको देखने के लिए मिलता है. Why mitosis process is very important for us and where we can found mitosis is happening. So the first and the most important significance of mitosis is that our body, हमारी बॉडी अच्छा डिप्लॉयड है हैप्लॉयड है? Our body is haploid or diploid?
Haploid.
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 We are diploid. We are diploids. Haploids are only gamete cells. Except gamete cell, all the cells of human body are diploid. And in the starting of this chapter, we have studied कि cell cycle में we need a growth. सबसे पहला and the most important topic was growth ना.
Yes, ma'am.
Yes, ma'am.
तो growth कैसे होगी? How will it happen? Growth will happen with mitosis only. तो it means हम लोग जितने भी organisms हैं, हमारे अंदर जो growth हो रही है, it is happening.
अरे भरी के दो.
Sha, mute yourself. तो हमारी जितने भी लिविंग ऑर्गेनिजम्स हैं, उन सब की ग्रोथ हो रही है किसकी वजह से? विद द हेल्प ऑफ माइटोसिस ओनली. ऑल डिप्लॉयड ऑर्गेनिज्म ग्रो विद द हेल्प ऑफ माइटोसिस. ठीक है. एंड माइटोसिस के अंदर वी हैव स्टडीड माइटोसिस क्या करता है आपका? नंबर ऑफ क्रोमोसोम्स रिमेन सेम इन पैरेंट सेल एज वेल एज इन डॉटर सेल. देयर इज नो चेंज ना. So it means by the help of mitosis, genetic complement also get same. Genetic complement भी same ही रहता है. Mitosis results in the production of diploid daughter cells बनता है with identical genetic complement. And जितना भी multicellular, all multicellular organisms growth is possible just because of mitosis. And if there is any type of damage in our body, हमारी बॉडी के अंदर किसी भी टाइप का डैमेज है, डैमेज इन आर स्किन, एपिडर्मिस इज द आउटर मोस्ट लेयर ऑफ स्किन. हमारी बॉडी का जो सबसे आउटर मोस्ट कवरिंग है दिस इज योर एपिडर्मिस. हमारी जो इंटेस्टाइनल्स की लाइनिंग्स है उसमें कोई डैमेज आ रहा है, हमारी ब्लड सेल्स के अंदर अगर कोई डैमेज आ रहा है, तो उन सब डैमेज को कौन रिपेयर कराएगा? माइटोसिस ओनली. हमारे सेल और साइटोप्लाज्म, सेल के अंदर न्यूक्लियस और साइटोप्लाज्मिक रेशियो को भी मेंटेन करने में who will help? Mitosis will help in maintenance of nucleus cytoplasmic ratio. Clear? The first one point. The growth of all diploid organisms occur just because of mitosis. Mitosis helps in the maintaining identical genetic complement in all daughter cells. Mitosis also helps in repairing of our epidermis, in our intestines, in our body cells as well as it also help to restore nucleus cytoplasmic ratio. As well as we all know plants के अंदर अनलिमिटेड ग्रोथ होती है नहीं होती है? प्लांट्स का ग्रोथ थ्रू आउट देयर लाइफ चलता रहता है. दे हैव अ कॉन्टिन्यूस ग्रोथ. तो वो कॉन्टिन्यूस ग्रोथ जस्ट बिकॉज ऑफ माइटोसिस ओनली. माइटोसिस की वजह से ही प्लांट शो इनडिटर्मिनेट ग्रोथ. डन ऑल 3 पॉइंट्स. So
Yes,
these
ma'am.
are the significance of mitosis. All clear, everyone?
Yes, ma'am.
Yes, ma'am.
Very good.
Yes, ma'am.
नेक्स्ट आ जाते हैं अब हमारा नेक्स्ट प्रोसेस वी हैव meiosis. Meiosis is also a division phase ना. Meiosis is also a division phase. Mitosis is a type of mitosis
No,
was
ma'am, it is not division phase. It is reductional division.
हाँ but है तो division ना. I'm not
Yes,
saying
ma'am.
equational है कैसे but है division ना.
Yes,
Yes, ma'am.
Exactly. So, if I say mitosis was equational division, in which number of chromosomes remain same, then just opposite to the mitosis, meiosis is reductional division.
yes.
Why we are calling it reductional division?
The number of chromosomes is reduced to half.
Very good. Number of chromosomes reduced to half in daughter cell. Let's suppose if in a parent cell we have 46 chromosomes, then in daughter cell how many chromosomes?
23.
23. We can write it 2n. Then in daughter cell how many?
And
And 2n is known as,
diploid.
and n is
Haploid.
haploid. हमेशा क्या होते हैं?
Gametes.
गेमीट्स होते हैं. इट मीन्स मियोसिस हम जब भी कराएंगे तो मियोसिस सिर्फ और सिर्फ किसके लिए होगा? जर्म सेल्स बनाने के लिए होगा. गैमीट्स बनवाने के लिए ही सिर्फ मियोसिस कराया जाता है, उसके अलावा कभी मियोसिस नहीं होता. मियोसिस हैपेंस ओनली ड्यूरिंग द फॉर्मेशन ऑफ गैमीट्स. Done?
Yes, ma'am.
वेरी गुड. सो विल स्टार्ट विद प्रोफेज. जैसे हमारा माइटोसिस में था, माइटोसिस के अंदर भी we have 4 phases: प्रोफेज, मेटाफेज, एनाफेज, टेलोफेज. हियर वी आल्सो हैव 4 phases. We are we are writing it at meiosis 1. Are you able to understand why we are doing it meiosis 1? Because meiosis are also of 2 type: meiosis 1 and then meiosis 2. Meiosis 1 and meiosis 2. So we are starting with meiosis 1. So meiosis 1 का जितना भी phases होगा उसको हम prophase 1, metaphase 1, telophase 1 ऐसे लिखेंगे.
Yes, ma'am.
Yes,
And
ma'am.
for meiosis 2 उसका जितना भी phases होगा उसको हम लिखेंगे prophase 2, metaphase 2, anaphase 2 and telophase 2. If somewhere it is written as anaphase 2 तो किसका phase होगा?
Meiosis 2.
Meiosis का.
Meiosis 2 है ना. It will be of meiosis 2. If somewhere it is written as only anaphase,
Mitosis.
mitosis होगा. तो ये डिफरेंशिएशन समझ में आया. सिर्फ एनाफेज, माइटोसिस. एनाफेज 1, meiosis 1. एनाफेज 2,
Meiosis 2.
exactly. So in the prophase, in the meiosis 1, prophase 1 is known as the longest phase because prophase 1 has 5 subphases. It has 5 subphases. The first one we have leptotene, then zygotene, then pectine, then diplotene, and then we have diakinase.
Diakinesis.
learn all these phases in a proper sequence. Same sequence where you need to learn these phases. The first one is,
leptotene,
second,
zygotene,
third,
pachytene,
fourth,
diplotene,
fifth.
dikinesis.
एक
Ma'am,
sequence में बोलो.
I have mnemonic for learning this.
बताओ
May I
निमोनिक.
लता जरा पानी दे दे.
सेम मैंने भी ऐसे याद किया. आपने इसको लेप्टो को लता बोला, मैंने इसको लीजा बोला हुआ है. लीजा जरा पानी दे दो. लेप्टोटीन लीजा, जाइगोटीन जरा, पेकाइटीन पानी दे दो. ये ट्रिक मैं आपको इसलिए नहीं, निमोनिक इसलिए नहीं बता रही थी बिकॉज यू ऑल हैव इश्यूज विद हिंदी. एंड अगर आप इतने हिंदी समझते हो तो हम लोग निमोनिक से याद करेंगे बहुत इजीली. ठीक है.
Yes,
Yes.
ma'am.
Okay then. So let's start with the first one phase which is leptotene. So we all know अब तक हमारा जो सेल था meiosis 1 से पहले कौन सी फेज से निकल के आया? G2 phase से. From G2 phase, cell will directly enter into which phase? Leptotene phase. है ना? You can write it here also. Cell starts with G1 phase, then S phase, then G2 phase, and then it will enter into
M phase,
leptotene. So in the G1 to G2 phase, genetic material is in which form?
chromatin,
Chromatin.
So just after entering the leptotene, what should be the first step? Condensation of
compact of chromosome,
compaction, or we can say condensation of chromosomes. So leptotene is the only phase in which chromatin will get converted into
Chromosome.
ठीक है. तो यू कैन रीड इट हियर. लेप्टोटीन स्टेज में क्या होता है? ड्यूरिंग लेप्टोटीन स्टेज क्रोमोसोम्स ग्रेजुअली स्लोली विजिबल होना स्टार्ट होते हैं. एंड क्रोमोसोम्स का जो एक कंडेंसेशन है ये पूरी लेप्टोटीन फेज में चलता रहता है. फर्स्ट फेज क्लियर. लेप्टोटीन इज अ वेरी स्मॉल फेज. देन वी हैव zygote. Do you remember what is zygote? Zygote formed by the fusion of male and female gamete.
Yes, ma'am.
Yes, ma'am.
जाइगोट फॉर्म बाय द फ्यूजन ऑफ टू सेल्स. दो सेल्स के फ्यूजन से बनता है. तो इस स्टेज का नाम जाइगोटीन रखा गया है. इट मींस कहीं ना कहीं यहाँ पे देयर शुड बी फ्यूजन ऑफ टू बॉडीज. दो बॉडीज का फ्यूजन हो रहा होगा यहाँ पे. दैट्स व्हाई वी आर कॉलिंग इट जाइगोटीन. सो विल रिमेंबर दिस फेज इन दिस वे ओनली कि इस फेज में क्या होता है? वी हैव क्रोमोसोम्स पेयरिंग करना स्टार्ट कर देते हैं. क्रोमोसोम्स स्टार्ट पेयरिंग टुगेदर and the process of pairing is known as synapses. And such paired chromosomes are known as homologous
Homologous.
Let's suppose we have a chromosome this. We have these two homologous chromosomes. These two chromosomes comes closer to each other and start pairing. इन दोनों क्रोमोसोम ने, let's suppose, can you see my video? ये मेरा hand is also a chromosome and this is also a chromosome. तो दोनों hands मेरे similar है बिल्कुल. Both two hands are similar, so we can call it homologous hands.
yes, ma'am.
अभी
Yes, ma'am.
my two hands are closing together. दोनों hands एक साथ आ रहे हैं. तो it means दोनों hands एक साथ आ रहे हैं. This process is
Synaptonymal complex,
like this process is synapses. ठीक है. अब आई वांट कि मेरे जो दोनों हैंड्स है ना आपस में स्टिक रह जाए. तो आई नीड अ ग्लू हियर जिसकी वजह से स्टिक रहेंगे. आई नीड अ ग्लू ना जिसकी वजह से स्टिक रहे आपस में, अलग अलग ना हो जाए.
Yes, ma'am.
बताओ.
Yes, ma'am.
That glue is known as synaptonymal complex. अब लाइन पढ़ो. इलेक्ट्रॉन माइक्रोग्राफ ऑफ दिस स्टेज, इस स्टेज का जब हमने इलेक्ट्रॉन माइक्रोग्राफ बनाया, ये इंडिकेट किया, इट शोज वहां पे कि क्रोमोसोम साइनेप्सिस किसकी वजह से हुआ, क्रोमोसोम्स आपस में पेयरिंग, लेट सपोज मेरा एक ही क्रोमोसोम है, एक ही क्रोमोसोम है, दोनों ने आपस में से पेयरिंग करना शुरू कर दिया, ठीक है, पेयरिंग किया, तो पेयरिंग किस वजह से किया, बिकॉज मेरे दोनों हैंड्स के बीच में एक ग्लू आ गया था जिसने आपस में अट्रैक्ट किया एक दूसरे हैंड्स को एंड आपस में चिपक गए, तो पेयरिंग किस वजह से हुआ, हैंड में बीच में ग्लू आया, दैट ग्लू इज साइनेप्टोनिमल कॉम्प्लेक्स, दो होमोलोगस क्रोमोसोम्स के बीच में साइनेप्टोनिमल कॉम्प्लेक्स बना जिसकी वजह से क्रोमोसोम्स क्लोजली आए एंड उन्होंने पेयरिंग करना शुरू कर दिया, डन। अब पेयरिंग करने के बाद ये मेरा एक हैंड नहीं बना ना, दो हैंड्स आपस में जुड़ गए हैं, तो वी विल कॉल दीज टू हैंड्स इज नोन एज बाइवेलेंट, दो क्रोमोसोम्स आपस में जुड़ गए हैं। लेट्स हैव अ लुक हियर, these are two chromosomes ना,
Yes.
Yes,
we'll
ma'am.
call it bivalent, एंड साइनेप्सिस कितनी क्रोमैटिड्स के बीच में हो रहा है, 1 2 3 4, देयर आर साइनेप्सिस बिटवीन 4 क्रोमैटिड, तो bivalent, that's why is also known as tetrad. Bivalent means 2 chromosome and tetrad means 4 chromatids. Clear?
Yes, ma'am.
Yes, ma'am.
So what is happening in zygotine? Like zygote. In the zygote, male and female gamete fuse together. In zygotine, two homologous chromosomes fuse together with the help of synaptonymal complex, and this process of fusion is known as synapsis. And after fusion, they will form a bivalent or tetrad. Okay? After finishing the zygotine, they will enter into which phase? Lisa, Zara, Pani. Pani means which stage?
Pachygyne.
Pectine. In the pectine stage, in the last stage, we were making bivalent or tetrad. In the pectine stage, these bivalent or tetrad start appearing clearly. We can see bivalent or tetrads under electron microscope. ठीक है, you can see in the diagram also. आप यहाँ पे डायग्राम में भी देख सकते हैं कैसे इनका साइनेप्सिस हो रखा है, एंड अब साइनेप्सिस के बाद वो क्या करेंगे, एंड लेट्स सपोज मेरे पास यहाँ पे ये साइनेप्सिस हुआ है, ठीक है, साइनेप्सिस के बाद क्या होगा ना, मेरा इस हैंड के अंदर जो जेनेटिक मटेरियल होगा और इस हैंड के अंदर जो जेनेटिक मटेरियल होगा वो आपस में क्रॉसिंग ओवर हो जाएगा, जैसे फिंगर्स क्रॉस कर रहे हैं, दो हैंड्स क्लोजली आए, देयर इज अ क्रॉसिंग ओवर, तो सेम चीज हैपन इन क्रोमोसोम्स। क्रोमोसोम्स के अंदर क्या है, क्रोमोसोम्स के अंदर डीएनए है एंड डीएनए के अंदर जेनेटिक इंफॉर्मेशन है, तो इसका मतलब दोनों क्रोमोसोम्स, बोथ टू क्रोमोसोम्स विल शेयर इट्स जेनेटिक इंफॉर्मेशन, एक दूसरे को जेनेटिक इंफॉर्मेशन शेयर करेंगे, एंड दिस प्रोसेस इज नोन एज क्रॉसिंग ओवर. You can read it here. This stage is characterized by appearance of recombination nodule. मतलब दो जो क्रॉस हमारे पास क्रोमोसोम्स थे will get recombined. उनके बीच में कॉम्बिनेशन बन गया है, एंड दैट साइट जहां पे क्रॉसिंग ओवर हो रहा है अगर कहाँ पे होगा, नॉन सिस्टर. डू यू नो व्हाट इज नॉन सिस्टर क्रोमैटिड्स? These two
Yes, ma'am.
chromatids are known as sister chromatids,
Sister chromatids.
and these two are known as
Non-sister chromatids.
Non-sister chromatids.
नॉन सिस्टर क्रोमैटिड्स. ठीक है, एंड क्रॉसिंग ओवर हमेशा किसके बीच में होगा, एक्सचेंज ऑफ जेनेटिक मटेरियल बिटवीन टू होमोलोगस क्रोमोसोम. क्रॉसिंग ओवर में एक एंजाइम इन्वॉल्व होगा, देयर विल बी अ एंजाइम एंड दैट एंजाइम इज रिकॉम्बिनेज. क्रॉसिंग ओवर में क्या होगा, जेनेटिक मटेरियल दो क्रोमोसोम्स के बीच में एक्सचेंज होगा. ठीक है, एंड आफ्टर फिनिशिंग द, जैसे पेकाइटिन स्टेज का एंड आएगा, आपका जो रिकॉम्बिनेशन है वो ऑलमोस्ट फिनिश हो जाएगा, और जो भी आपका क्रॉसिंग ओवर हुआ वो बिल्कुल कहाँ पे आ जाएगा, एंड पे आ जाएगा. इट विल लुक लाइक दिस, यहाँ पे एंड पे आपका ये आपका क्रॉसिंग ओवर स्ट्रक्चर है. दिस इज योर क्रॉसिंग ओवर. आर वी डन विद पेकाइटिन स्टेज? पेकाइटिन में
Yes,
सबसे
ma'am.
पहले क्या हुआ, क्लियरली विजिबल हुआ बाइवेलेंट और टेट्राड, उनके बीच में क्रॉसिंग ओवर इज द प्रोसेस ऑफ एक्सचेंज ऑफ जेनेटिक मटेरियल बिटवीन टू नॉन सिस्टर क्रोमैटिड. इन द क्रॉसिंग ओवर देयर इज an enzyme which is known as recombinase, and recombination process will finish at the end of pegiding. After pegiding we have which phase?
Diplotin.
Diplotin.
In diplotin what will happen? That one glue साइनेप्टोनिमल कॉम्प्लेक्स जो हैंड्स के बीच में एक ग्लू है, तो अभी ग्लू क्या होगा, धीरे धीरे, लेट्स सपोज जस्ट फोकस ऑन माय हैंड, अभी क्या हो रहा है, साइनेप्टोनिमल कॉम्प्लेक्स स्टार्ट डिसॉल्व होने लग रहा है. और ये सिर्फ कहां से डिसॉल्व नहीं होगा, ओनली एट दैट पॉइंट जहां पे क्रॉसिंग ओवर हो रहा है. क्रॉसिंग ओवर इज हैपनिंग ओनली एट माय फिंगर्स ना, तो नीचे से पूरा साइनेप्टोनिमल कॉम्प्लेक्स हैज डिसॉल्व्ड. Now we have synaptonimal complex only at my points where crossing over is going to happen. तो यहाँ पे कौन सी shape structure बन रहा है? Which shape is making? X shape. And this X shape structure is known as? This X shape structure is known as Guy's beta. तो डिप्लोटीन के अंदर क्या होगा? डिप्लोटीन, D for डिप्लोटीन, D for dissolution. डिसोल्यूशन ऑफ द साइनेप्टोनिमल कॉम्प्लेक्स. हर जगह से साइनेप्टोनिमल कॉम्प्लेक्स हट गया है एक्सेप्ट एट द साइट ऑफ क्रॉसिंग ओवर. जहां पे क्रॉसिंग ओवर हुआ था उस साइड के अलावा हर जगह से इट हैज ऑलरेडी डिसॉल्व्ड. एंड डिसॉल्व होने के बाद वी विल मेक अ एक्स शेप स्ट्रक्चर. दैट एक्स शेप स्ट्रक्चर इज नोन एज Guy's beta. In some oocytes of some vertebrates, we all know very well vertebrates क्या होता है. वर्टिब्रेट्स की जो oocytes होते हैं उनके अंदर जो डिप्लोटीन होता है मंथ्स और ईयर्स तक भी रहता है. डिप्लोटीन को फिनिश होने में टुक्स मंथ्स टू ईयर्स. आफ्टर डिप्लोटीन वी हैव व्हिच फेज?
Dikinesis.
Dikinesis. What will happen in the dikinesis? That X shaped structure कहाँ पे आ जाएगी? वो बिल्कुल टर्मिनल पोजीशन पे आ जाएगा. That X shaped structure get at the terminal point. And this is the initiation of dikinesis. After finishing the terminalization, all the cell mitotic spindles start appearing, cell organelles start dissolving, nuclear envelope also start, just like mitosis. बिल्कुल mitosis के जैसे सेम हो रहा है. तो फिर से एक बार रिकैप करें. अच्छा, कोई रिकैप कराना चाहेगा? All 5 phases: leptotene, zygotene, pectine, diplotin, digon, in your own words. Yes, Harshita.
Ma'am, in meiosis 1 phases, prophase 1 we have seen it is the longest and complex phase. In prophase there are 5 phases. First is leptotene. In
Okay.
this there is a compaction of chromosome and nuclear membrane and nucleolus is also present. In zygotene there is pairing of homologous chromosome that due to which the pairing is happening. That glue is known as synaptonimal complex, and this process is known as synapsis, and it is get converted into bivalent. The third process is pectine. It also forms tetrad, and it is the crossing over between 2 chromosomes, and that crossing over is known as recombination nodule. Fourth phase is diplotene. It is the dissolution of synaptomal complex, and it also gives rise to X-shaped structure. It is the result of crossing over but not cause of crossing over. And in example we have seen that in oocyte of some vertebrated, it lasts for a month and a year. It is known as dictotine. And last phase is dikinesis. In this phase nuclear membrane and nucleolus disappear, and chiasmata move towards the end of chromatid. It is known as terminalization. That's it.
Very good, Harshada. Then after the finishing the dikinesis, the cell will enter into which phase? Cell will enter into which phase? Obviously, metaphase one. So, who will tell me what happens in metaphase of mitosis? Anyone else? Irfan, Anusha, Shikha. Yes, Aditya. आप बताइए.
Ma'am, they align at the equatorial plate.
Yeah.
The bivalent chromosomes align at the equatorial plate, ma'am.
Okay. All the chromosomes get align itself at the equator plate, and that equator plate is known as metaphase plate. So here is also same. Here is also same. After finishing the dikinesis, the cell will enter into metaphase 1, in which all chromosomes get align itself at equator and their kinetochore get attached with spindle fibers. And after finishing the metaphase 1, cell will enter into which phase?
Anaphase
Anaphase
1.
1.
Anaphase 1.
Anaphase 1. What will happen in anaphase 1?
Chromosomes get separate, ma'am.
Yeah, this is the main difference here. In the mitosis, centromere split. In the mitosis, centromere split, because we need to divide the chromosomes in equal number. If the parent cell has 4 chromosomes, then the both daughter cells should have 4 chromosomes. But here, there is no such case. If we have 4 chromosomes, we can give 2 chromosomes to one cell and 2 chromosomes to another cell. So there is no need of separation of chromosomes. Is there any need for separation of chromosome?
No, ma'am.
So
No, ma'am.
in the anaphase 1, there is no splitting of centromere. There is no splitting of centromere. If the cell has, let's suppose, we have 4 chromosomes. We are in this phase. Okay? And then after finishing the metaphase, what will happen? 2 chromosomes will move at one pole and 2 chromosomes will move at
Another pole.
another pole. So it means in anaphase 1, only chromosomes will
Separate.
move. Chromosomes will move to opposite poles. Chromosomes will move to the opposite
Opposite
poles.
pole.
There should be no centromere splitting. This is known as anaphase 1. And after finishing the anaphase, cell will enter into which phase?
Telophase
Telophase
1.
1.
Telophase 1. And telophase 1 is just opposite to the, telophase 1 is just similar to telophase. There is decondensation of chromosome. Reappearance of all cell organelles, reappearance of
Nuclear
nuclear
membrane.
envelope.
Nuclear membrane.
This is
Nucleolus.
telophase 1. And after finishing the telophase, cell will enter into which phase?
Meiosis 2.
Cytokinesis.
Meiosis 2.
Firstly, it will go into cytokinesis.
Cytokine.
सबसे पहले cytokinesis होगा, then it will enter into
Meiosis 2.
Meiosis 2.
meiosis 2. And meiosis 2 is just similar to
Mitosis.
Meiosis 1.
meiosis 1. In meiosis 2, number of chromosome remain same. So it means in anaphase 2, should be splitting of centromere. Right or not?
Yes, ma'am.
Yes, ma'am.
There should be splitting of centromere. So this was all about meiosis 1 and meiosis 2. Can you tell me the difference between mitosis and meiosis? बताओ. Raise your hand if you know the answer. Yes, Anusha, tell me. जितना आपको पता.
Mitosis is a equational division. Meiosis is a reductional division. Mitosis take place in somatic cells. Meiosis take place in germ cells. Mitosis produce 2 daughter cells. Meiosis produce 4 daughter cells. Synapsis does not occur. Synapsis takes place.
Exactly. We can say the first difference is mitosis is Equational
Well,
division. But meiosis is
Reductional division.
reductional division. If I have a cell of 2n ploidy, then after mitosis, I will get two cells of same number of chromosomes. But
Yes,
in
ma'am.
meiosis, what will happen? If I'll start with meiosis 1, meiosis 1 में क्या होगा? I'll get two cells of NN.
1N.
Then after these 2n, meiosis 2 will occur. Then meiosis 2 में I will get another two cells of which ploidy? Are you able to understand this chart?
yes, ma'am.
Yes, Yes, ma'am. ma'am.
है ना? And if we can say, if we can also differentiate it on the basis of anaphase. We can also differentiate it on the basis of anaphase, na?
Yes, ma'am.
Because here in anaphase, only in anaphase, centromere split. Anaphase 1 not split. And in anaphase 2,
Split. Centromere split.
again split. Very good. So this is the basic difference between mitosis and meiosis. Can we give a difference between meiosis 1 and meiosis 2? Obviously, we can give. Meiosis 1 is reductional division. And meiosis 2 is
Equational division.
Equational division.
equational division. We can also differentiate it on the basis of anaphase.
Anaphase.
Anaphase के basis पे we can also differentiate it. Let's practice some questions. Now we have first question. Acha, before solving this question, can you please mark your attendance? I'm just sharing it in your group. Give me a second. Firstly, mark your attendance, then we will practice some questions. I have shared in the chat section. If you are done with your attendance, give me a thumbs up. And try to fill your proper details here. Fill your correct mobile number and correct DOB, what they are asking. Everything should be correct unless your attendance will not mark. And if you have 90% attendance, only then you will be able to get study materials. Only those students who have 90% attendance, those students will get hard copies of study material. Harshada, firstly mark your attendance, then we will solve this question. Everyone, hurry up. If you are done with your attendance, then start solving this question. It's a very simple question. Anusha is saying option B. Okay. Davisha is also saying option B. आर्य बच्चों Okay. So let me tell you the correct answer. The correct answer is in the zygote stage, what happens?
Synapses.
Synapses. Very good. So A should be fourth, and the correct answer is B. So very simple. Next question. Arrange the following events of meiosis in the correct sequence. Very good, Harshida. Harshida is saying B. So I guess that should be from previous question. Arya Bachu, the first should be crossing over your disappearance of nuclear earth. Obviously, first should be synapses. Correct answer is D. Why should not C? Because the first step is synapses. Synapses ke baad we have crossing over and then terminalization. Terminalization of Chias beta occurs in diplotin stage. Dissolution and then terminalization diakinesis में होता है. So that's why the correct answer should be D. Agree?
Yes, ma'am.
Yes, ma'am.
Next question. Friends, have you marked your attendance? Everyone, just raise your hand if you have marked your attendance. Only 5 students. Rest 6. Okay. 7. Others? Baki students, please mark your attendance. 3 students left. Ma'am, it would be C. Okay, let's check the answer. Synthesis of RNA and protein happens in G2 phase, so B should be third. So accordingly, B and D can be the correct answer. Centromere do not split, but chromatid move toward opposite poles. This is your anaphase 1, sorry. This is anaphase 1. So D should be fourth. The correct answer is D. Agree everyone?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Devisha is saying B. Devisha, why are you saying B? Okay then. Next question. Question is already asked in NEET 2019. You cannot ignore this. Here, everyone is saying correct answer is B. Crossing over take place between two chromatid in which non-sister chromatids of homologous chromosomes in pectiding stage of prophase 1. Very well done, Bachu. Very well done. The correct is B only. Very nice. So let's have a quick recap what we have done in today's class. Just tell me what we have learned in today's class. कुछ ऐसा नया जो हमने आज सीखा है.
All 5 phases of meiosis 2.
वीजा, जरा, पानी, दे, दो. What happens in leptotene?
Chromosomes become gradually visible.
Compaction.
Compaction of
happens in-
chromosome.
The zygotine.
Pairing of homologous chromosome.
Synapses.
Synapses. What happens in pectidine?
Crossing over.
Crossing over.
Divalent. Yeah, we can say bivalent. And
Dissolution of synaptomal complex.
crossing over in the D.
Dissolution.
Dissolution and in dikinesis.
Termination.
Disappears.
Terminalization.
Terminalization. Well, we have done these 5 phases. And if someone asks difference between anaphase 1 and anaphase 2, we can give the correct answer
Yes, ma'am.
on the basis of splitting. Splitting occurs, splitting do not occur, splitting occurs. Done?
Yes, ma'am.
Yes, ma'am.
Okay then. For homework, you can check your group. In the group, I have already shared a 36 years, 33 years MTG book. You can refer that one book for solving the more PYQs. And in the next class, what we will do in the next class, we'll start with a new chapter. This is your respiration in plant. Or anything else? Do you want to, any other chapter do you want to start according to your preference, or it is good to go? कोई वो चैप्टर है जो आप स्टार्ट कराना चाहते हैं? एनी डिफिकल्ट चैप्टर डू यू फाइंड कि हाँ सबसे ज्यादा
Animal
डिफिकल्ट है?
kingdom.